So Lexi and I are doing the Phi-X 174 phage.
So the Phi-X 174 is a bacteriophage that it's a virus that infects and replicates within bacteria, and Phi-X basically infects E. coli. It's a single-stranded DNA genome, which is very rare. It's a positive sense single-stranded DNA. And it goes through rolling circle replication.
So basically, just an overview of how it infects E. coli is it binds to the lipopolysaccharides on the outside of E. coli because, as we know, E. coli is gram-negative, and so it binds to those lipopolysaccharides, and it will inject and replicate its DNA inside the host. And so there you can see just a diagram. The phage is that like blue thing, and then it's attaching to the lipopolysaccharides, and there you can see through the H tube it injects its single-stranded DNA into the host.
So we discussed that it goes through rolling circle replication, but what is that? So rolling circle replication is a one direction replication of circular DNA. It Phi-X specifically starts out as a positive sense single-stranded DNA. So it needs to do that synthesis of a negative strand complementary to the positive strand. So after doing that, the double-stranded DNA is nicked at a site to create an unraveling. So the unnicked DNA is the leading strand, so the template. And then the nicked strand progressively peels off to create another single-stranded DNA. And then from there, it can go on to make mRNA and proteins.
So this is the structure and genome of it. It's an icosahedral capsid, and it's tailless. So most like viruses and like bacteriophages, they kind of look like that spider, that goes and attaches. But as you could see from one of the previous slides, it's that like capsid, like capsule kind of looking thing instead of like the spider looking thing. So that's what tailless is. And so it is a circular single-stranded DNA. And the first evidence of overlapping genes, so it has 11 genes and 8 of them are overlapping. So you can kind of see that towards the top with like E, D, C, K, B, like those genes, those are the overlapping genes. And so the nucleotide sequence partially overlaps with the nucleotide sequence of another gene, and that's what you're seeing in that diagram. So a nucleotide sequence can contribute to the function of more than one gene. And again, that's what you see in that. And it's possibly due to the fact that it's single-stranded instead of double-stranded. So it just kind of compensates everything. So. And then here's some historical background. So Arthur Kornberg in like 1959 was the first to like synthesize in a test tube like the features of a natural virus through purified enzymes. And this was a huge step forward in synthetic biology. And then Fred Sanger in 1977, he used this bacteriophage to make the first DNA like base genome to be sequenced.
So this is just my personal application of Phi-X. In our lab, we use Phi-X as a viral tracer. So when we're doing experiments that we want to test like where virus spreads, like in the air or on surfaces, rather than using like an infectious virus, we will use Phi-X and then swab areas where we expect the Phi-X to be after, like putting on our hands and like seeing where it goes. We will aseptically plate onto TSA with an E. coli host. And then the entire plate, as you can see in the picture, is covered in E. coli. And those clear black plaques show where the Phi-X, quote unquote, ate the E. coli. So then we can like numerically see how much Phi-X was on that sample. And yeah, we love Phi-X.